section 8.4 and 8.5 we're going to do together because transcription and translation kind of go hand in hand. You have to know the first one before you can do the second one, and a lot of the terms that I'm going to use in the first process are carried over into the second process. So transcription is when you transcribe something or you write something down or make a copy of something, versus translation is we convert it to a different language. RNA is a new piece of our genetic code that we have yet to talk about, but it stands for ribonucleic acid, and it carries our DNA's instructions based on using the DNA to make the RNA, which in turn allows us to make the protein. That in itself is known as the central dogma, and it allows us to understand how information flows in one direction from DNA to RNA. When we're talking about the central dogma, there are three processes that are included. First, replication, which is something we're building upon from section 8.3, but then to get from DNA to RNA, we use a transcription process, and then from RNA to protein, we use a translation process. How do they differ between RNA and DNA? RNA has a ribose sugar versus DNA has a deoxyribose sugar. RNA uses the nucleotide uracil, and DNA has thymine, so A is now going to pair with a U in RNA, and a T is not going to exist when we're talking about RNA. RNA is also one strand versus DNA would be a double strand. And that's what uracil would look like. So for transcription to take place, there are three major processes that have to happen, and they're very similar to that of DNA replication. In the first process, a start codon is being searched for by this transcription complex known as RNA polymerase. RNA polymerase is going to open up the DNA strand and expose those nucleotides so we can use them as a template to make our RNA strand. The second step, just like in replication, free-floating nucleotides have to pair up to the DNA strands. We're going to use one side of DNA, whether it's this side or this side, it doesn't matter. But the free-floating nucleotides are going to begin to pair to one side of the opened up DNA in order for the RNA strand, which is this red strand, to be created. In the third step of transcription, the RNA strand is going to break off and head towards the ribosome. This whole process is still within the nucleus because that's where DNA is found. Once the RNA strand breaks off and heads towards the nucleus, the RNA polymerase enzyme is going to break apart and DNA is going to seal back together with its pairs. There's three types of RNA that are made during the transcription process. First, you have mRNA, or messenger RNA. A messenger is someone who carries a message. So our messenger RNA is going to take the message that was created in the nucleus to the ribosomes where a protein can be formed, because that's our end result as far as transcription and translation, protein synthesis or making of a protein. Ribosomal RNA is found within the ribosomes, and transfer RNA is the tRNA that's going to be found in the cytoplasm that brings amino acids, which are the components that make up a protein, the monomers to the polymers, if you remember chapter 2, that bring the amino acids to the ribosome so we can make our proteins. That is 8.4. Just an extra slide for you to take a look at in comparing replication and transcription. They're very similar processes, except replication is making new DNA versus transcription is making copies of a gene into RNA so we can use it to make a protein. For section 8.5, translation is going to convert that message that was created by our mRNA strand into a polypeptide, which is our protein sequence. To do that, we need codons. A codon is a three-letter sequence that codes for a particular amino acid on page 244 in your textbook. And that chart will be coming up shortly in the next few slides. A codon, you need 3 letters because there are 20 essential amino acids that are needed to make all the proteins we have in our body. AUG happens to be a start codon. There's only one start codon in the chart, and there's 3 stop codons. When the process is happening as far as translation, it's going to continue until a stop codon is reached. And this is the chart I was talking about. With how this chart is read, your first letter, so that on the previous slide it talked about AUG. If I go back AUG is the first sequence that we're looking at. That's our codon. So we would look for the A along this first column here. We would look for the U across the top, and we would look for where the G meets up. So we have A, U, and G coming across. Our amino acid would be methionine, which happens to be the start codon. You could look up CCU, C, C, and a U come together right here. That would be proline, which is another type of amino acid that is going to be found in our chains to create our proteins. At any point you reach a stop codon, which would be UAA, UAG, or UGA, the sequence is going to stop, the protein is going to break off and go carry out its function. The reading frame is the series of 3 non-overlapping nucleotides that are read in order to create our amino acids. So our reading frame here would create the first amino acid, this one would create the second amino acid, this one would create the third amino acid, this one would create the fourth amino acid. I'm going to have to pause for just a second.